In today's class, we will see about cell cycle and cell division. We know that the dead and worn out cells are to be replaced. It means new cells have to come in their place, and this occurs by cell division from similar cells throughout the body. And this rate of growth of cells differs in different tissues based upon their function and based upon their activity. And this is a very carefully regulated process to maintain the normal number of cells that are present in any tissue or organ. We know that all the cells have 46 chromosomes, or which we call them as 23 pairs of chromosomes. But when these cells divide, the daughter cells will have 46 chromosomes each, and this will be attained during the time of cell division. Means the chromosome number will be doubled and it will be distributed to both its daughter cells.
Cell cycle is a series of well-orchestrated steps including cell growth, duplication of genetic material, and cell division, usually resulting in two identical cells. In other words, a cell passes through various steps in order to grow, copy its DNA, and finally split into either 2 or 4 new cells. This is cell cycle. And what exactly is cell division? As the name suggests, cell division is the process in which a single parent cell divides into either 2 identical cells or 4 cells, each with half the number of chromosomes. So can we say that the process of cell division is a part of the cell cycle? That's right. That's how it is. Now tell me one thing. Do all the cells divide in the same way? Just think about it. The answer is a definite no, right? Every cell in our body has a different tendency to grow and divide. To be precise, some cells divide throughout their lifetime, while others do not divide once formed. For example, our skin cells have a high turnover rate due to constant wear and tear and divide very frequently. On the other hand, our nerve cells do not divide once formed. Now getting back to the cell cycle, as the name suggests, the process occurs in phases or stages, the series of steps that repeat in cyclic manner. But for our convenience, we majorly divide a cell cycle into 2 phases. The first phase is called the interphase, and the next is the M phase, which represents either mitotic or meiotic phase. Let's have a quick look at these phases. The interphase prepares the cell for cell division, hence it's also called the preparatory phase. In this phase, the cell grows and makes a copy of its genetic material, the DNA to be precise. On the other hand, the M phase is the dividing phase of the cell cycle. It carries out the task of actual cell division. After going through a series of steps, the cell first divides the nucleus into 2 sets, followed by the division of the cytoplasm. This gives rise to 2 identical daughter cells. And this process of cell division is called mitosis. Similarly, instead of once, a parent cell divides twice to give 4 daughter cells, each with half the amount of genetic material compared to the parent cells. This type of division is called meiosis, the one useful in the production of gametes.
There are 2 types of cell divisions. One is mitosis and the other is meiosis. We have seen in those daughter cells where the number of chromosomes are identical to that of their parent cell, such division is called as mitosis. Whereas in the formation of gametes such as ova and spermatozoa, which occurs by a process called as meiosis, the number of chromosomes will be half to that of their parent cell, and this process of cell division, it is called as meiosis. It means at the end of mitosis, the daughter cell will have similar number of chromosomes as that of parent cell, whereas at the end of meiosis, the daughter cell will have half the number of chromosomes from its parent cell, and hence these gametes are called as haploid cells because they have only 23 chromosomes, whereas the cells which are formed after mitosis, these are called as diploid cells because they have 46 chromosomes or 23 pairs of chromosomes.
The interphase lasts much longer compared to the M phase. You will be astonished to know that the interphase occupies more than 90% of the total time required for one cycle. And only the remaining less than 10% of the time is needed for the M phase to get the new cells formed. So all the important functions like growth, duplication of genetic material, synthesis of newer compounds and organelles, all of these are carried out in the interphase. It is believed that the cells take up approximately 24 hours to complete one cycle. However, this duration can vary from organism to organism and from one cell type to the other. Later, at the end of the cycle, the new cells produced can start the exact same process all over again. That's the reason why it's called a cycle, as it does not follow a linear pathway.
Now, let's see about these two different types of cell division, that is mitosis and meiosis. First, let's start with mitosis. Mitosis is a cell division which occurs in almost all the cells in the body except those cells which are involved in production of gametes. And this sequence of events which occur during the cell division can be classified or called into cell cycle. So, cell cycle is the duration between two different cell divisions of a particular cell. And this cell cycle, it consists of two phases which can be seen under light microscopy. Here, the two phases are, one is interphase and the second is mitosis, or it is also called as M phase. So these are the two phases, interphase and M phase. Of these two phases, interphase is the longer phase than that of mitosis phase. And during this interphase, it prepares the cell for cell division. And this interphase once again occurs in three stages. They are first gap phase, S phase, and second gap phase. First gap phase, S phase, and second gap phase. These are the three phases of, or we can call them as three stages of interphase. Let's see them in detail. The first gap phase. This phase is also called as G1 phase. In this phase, there occurs growth in size of the cell, so which leads to increase in volume of the cell. And this G1 phase is also the longest of the three phases, and this might not last throughout the cell cycle, because in some cases the cells might enter into a resting phase called as G0 phase. If the cell does not enter into the resting stage, then it goes into the next phase, that is S phase. And during the G1 phase, the cell will be carrying out its normal functions such as secretion or absorption. The second of interphase is S phase. S phase is also called as synthetic phase, and this is the phase in which the DNA will be replicated. It means the number of chromosomes will be doubled. The 23 pairs of chromosomes will become 46 pairs of chromosomes, so that the DNA is sufficient for 2 daughter cells. And after this phase comes the third phase, that is second gap phase, and this is also called as G2 phase. There is further growth and preparation for cell division. So these are the 3 stages of interphase: first gap phase, S phase, and second gap phase.
Is an image that depicts the complete interphase in general. As we can see, the phase has 3 stages. Let's begin each stage step by step. The first stage of the interphase is the first gap phase, abbreviated as G1 phase. This is the first phase occurring in interphase, hence the name first gap phase. It corresponds to the interval between the M phase of the previous cycle and the S phase of the current cycle. Since this is the first stage of the interphase, that is, the first stage of the preparatory phase, and the phase which precedes synthesis of DNA, what do you think will be the activities of the cell? Just think about it. Well, as the cell is metabolically active, it grows in size in the G1 phase. All the organelles duplicate in this phase, and the building blocks which will be required in later stages are made by the cell. It is important to note that the G1 phase does not involve DNA replication. Now let's move to the second stage of interphase, which is the synthesis phase, abbreviated as S phase. As the name suggests, there will be synthesis of something. And what will that something be? The most crucial part of each cell, that's the nucleus, along with the genetic material within, will get synthesized, that is, duplicated. So the S phase takes up the charge of the DNA replication. Now, along with the nucleus, there is one more important organelle that gets duplicated in the S phase. Do we know this bundle of threads present in the cell? That is called the centrosome. This bunch contains smaller organelles called the centrioles placed within, usually at right angles. They are made up of microtubules which help in releasing the threads required for separating sister chromatids later. We will have a detailed look at this process in our upcoming videos. For now, all we need to know is that these threads are important for the cell division. And for this reason, duplication of the bundle containing these threads is also important. This happens in the S phase. So the S phase takes up the responsibility of duplicating the contents inside the nucleus and the centrosome present in the cytoplasm simultaneously. What next then? The next step in the process of the interphase is the second gap phase, abbreviated as G2 phase. This phase allows the cell to grow more. Along with the enlargement in the cell size, the cell also makes more proteins and organelles. The most important function of this phase is that it prepares the cell for mitosis. This phase helps the cell to prepare for the actual cell division in the M phase. So now, are we set to study the next phase? That is the M phase? No, not yet. We are yet to study one more intermediate phase. A stage which can be entered post the M phase of one cycle and before the G1 phase of the next cycle. There is a phase called the G0 phase in between. So why didn't we talk about this phase in the beginning before the G1 phase? Well, the cells may or may not opt to enter this phase. The cells may enter the G0 phase or may directly enter the G1 phase after the cell division. And what factor decides this? Well, there are several intrinsic and extrinsic factors like resource availability or nutritional deprivation which can force the cell to enter this G0 phase. But what exactly happens in this phase? This phase was classically thought to be only a resting phase, as the cell is metabolically active but does not show tendency to divide further. However,
after this interphase comes the mitosis phase, that is also called as M phase. And this can also be categorized, or we can say divided into 4 phases. They are prophase, metaphase, anaphase and telophase. These are the 4 stages of mitosis or M phase. Now let's see them in detail. In the prophase, pairs of identical chromosomes are tightly coiled with each other at centromere, leading to formation of 46 pairs which are tightly coiled at their centromeres. And during this phase, mitotic apparatus will appear. Mitotic apparatus consists of centrioles, 2 in number, and mitotic spindles. This centrioles and mitotic spindles are formed from the microtubules. And these 2 centrioles migrate to 2 ends of the cell and the nuclear envelope also disappears. The next stage is metaphase. And in this metaphase, all the paired chromatids will align in the center of the cell. And this is regulated by the mitotic apparatus. And in the third phase, that is anaphase, the centromere splits and the mitotic spindles will contract. This leads to pulling of the chromatids into each end of the cell, and this occurs due to contraction of this mitotic spindles. Whereas in the fourth phase, that is telophase, where the mitotic apparatus will disappear and the nuclear envelope also starts to reappear. Following the telophase is cytokinesis, and during this phase, the cell organelles, cytosol and plasma membrane, they split so that this can lead to formation of 2 identical daughter cells. This is mitotic type of cell division.
Prophase, metaphase, anaphase and telophase were the phases of the karyokinesis, a part of the dividing M phase. This is then followed by cytokinesis, which results in the division of the cytoplasm. Let's have a quick revision of all these phases. We are familiar with the events that the cell undergoes in this phase. The chromatin condenses to form the classical chromosome structure, having 2 sister chromatids. We very well know the chromatin is nothing but the DNA associated with the proteins. So condensation of this chromatin begins in the prophase. And what happens next? Correct. The centrosomes move apart and begin releasing mitotic spindles. The nuclear membrane and the nucleolus also start disappearing, which marks the breakdown of the nucleus. After going through these changes, the cell enters the metaphase. Can you tell me the most important change occurring during this phase? That's right. Formation of the equatorial plate. The chromosomes align at the equator, forming the unique metaphase plate. Along with this, the cell undergoes many other changes as well. The centrosomes reach the opposite poles. Also, the mitotic spindle attaches to the kinetic cores of the respective sister chromatids. All these changes prepare the cell to enter the next phase of the karyokinesis, that is, the anaphase. Do you remember that anaphase is the shortest and most rapidly occurring phase in the complete karyokinesis? Yes, in this phase, the sister chromatids separate and begin their migration to the opposite poles of the cell. The cell then enters the last phase of the karyokinesis. We know this phase well, it's called the telophase. Here, the cell, 2 nuclei with daughter chromosomes are formed in the single cell. And this is possible as the phase carries out all the tasks reverse of what happened in the prophase. The chromosomes decondense to gain the relaxed string-like chromatin structure. And are we left with something else too? That's right, the nuclear membrane. The nuclear membrane and the nucleolus, which disappeared during the prophase, now start reappearing. So now the cell is all set for the last phase of the mitosis, that is cytokinesis. The phase of cytokinesis divides the cytoplasm of the cell. There is a formation of a cleavage furrow in the cell membrane which extends up to the core. And this results in the formation of two daughter cells. These two daughter cells are genetically identical to each other. In other words, they have an equal number of chromosomes in the nucleus.
Now, let's see the other type of cell division, that is meiosis. We have seen in mitosis, it leads to formation of two identical daughter cells with the same genetic material as that of parent cell, whereas this does not occur in the case of meiosis. And meiosis is the final step which is involved in the production of gametes. Unlike mitosis, meiosis also occurs in two different stages, or it undergoes two different cell divisions. That is, the first meiotic division and the second meiotic division. And at the end of meiosis, it leads to formation of four different daughter cells, which will have only haploid number of chromosomes, that is only 23 chromosomes. As these gametes are intended to fuse with other gamete to form a diploid zygote, and hence these are the two differences in meiosis when compared to that of mitosis. Meiosis occurs in two stages, that is first meiotic division and second meiotic division, and at the end of this second meiotic division, it leads to formation of four haploid daughter cells, and all are genetically different from each other and also from the parent cell.
This is an exceptional phenomenon which produces four daughter cells, each with half the number of chromosomes compared to the single parent cell. You heard me right. The process gives us four new cells, unlike in mitosis that produces only two new cells. And what could be the reason for this? Why do we need four daughter cells, and that too with half the number of chromosomes? Just think about it. Sexual reproduction involves both the parents, or two cells, one from each parent to be precise. Now, if there are two cells fusing, it's obvious that the number of chromosomes should be half in both the cells, so that the fused cell will carry the normal chromosome number. Now let's take it the other way. We know that the human chromosome number is 46. So, if there are two human cells fusing to give us an embryo, then the newly fused cell will carry 46 plus 46, 92 chromosomes. This is simply not possible in nature. So in order to maintain the chromosome number of species, it is necessary that the two fusing cells contain half, that is 23 chromosomes in number. So in order to maintain the number of chromosomes in the zygote, the chromosome number in the fusing cells need to be halved. Also, the chances of fertilization increase with more number of cells. Hence, instead of two, making four cells is preferable in nature. This is the reason why meiosis gives us four daughter cells with half the number of chromosomes. Now, since the process yields us cells with half the chromosome number, it's also called as reductional division. Now that we've got introduced to the process, let's glance at the definition once more, but this time with respect to the chromosome number. Meiosis is a process that gives rise to a haploid cell from the diploid one. Now you must be wondering, what is a haploid cell and a diploid cell? Well, haploid cell is the one that has a single set of chromosomes, while diploid cell has two sets of chromosomes. That means a diploid cell has chromosomes in double doses, while a haploid cell has chromosomes in a single dose. So the cells in our body with 46 chromosomes are all diploid, while the reproductive cells are haploid with 23 chromosomes. Now tell me, which are these reproductive cells that we've been talking about so far? Cells with half the number of chromosomes? Let's find the answer in our next video.
Now let's see what happens in the first meiotic division and the second meiotic division. In the first meiotic division, before this occurs, there occurs a replication of the DNA. It means the number of chromosomes will be doubled, and this leads to formation of 4 genetically different chromatids. Because these chromatids, which are tightly coiled with centromeres, can exchange the genes, which leads to formation of different chromatids with different genetic material from their parent cell. And this process of exchange of genes between the chromatids is called as crossing over. This crossing over leads to exchange of maternal and paternal chromosomes among themselves. So this leads to formation of 2 daughter cells. The first stage of meiotic cell division, or the first meiotic division, will lead to formation of 2 daughter cells, and these 2 daughter cells also will have genetically different chromosomes when compared to their parent cells. And once again, this cell division in first meiotic stage, it occurs under the influence of mitotic spindle, which leads to pulling of chromosomes to 2 ends of the cells and following by cell division. And this is followed by second meiotic division. In second meiotic division, once again there occurs splitting of these chromosomes from the 2 daughter cells that are formed after first meiotic cell division. This leads to formation of 2 daughter cells from each daughter cell of first meiotic division. With this leads to formation of 4 daughter cells, and all the 4 daughter cells will have entirely different genetic material when compared to that of their parent cells of first meiotic division, and they also will be entirely different from the parent cell before the first meiotic division. So at the end of meiosis, it leads to formation of 4 daughter cells, and these 4 daughter cells are having only 23 chromosomes.
This is an exceptional phenomenon which produces 4 daughter cells, each with half the number of chromosomes compared to the single parent cell. You heard me right. The process gives us 4 new cells, unlike in mitosis that produces only 2 new cells. And what could be the reason for this? Why do we need 4 daughter cells, and that too with half the number of chromosomes? Just think about it. Sexual reproduction involves both the parents, or 2 cells, one from each parent to be precise. Now if there are 2 cells fusing, it's obvious that the number of chromosomes should be half in both the cells, so that the fused cell will carry the normal chromosome number. Now let's take it the other way. We know that the human chromosome number is 46. So if there are 2 human cells fusing to give us an embryo, then the newly fused cell will carry 46 plus 46, 92 chromosomes. This is simply not possible in nature. So in order to maintain the chromosome number of species, it is necessary that the 2 fusing cells contain half, that is 23 chromosomes in number. So in order to maintain the number of chromosomes in the zygote, the chromosome number in the fusing cells need to be halved. Also, the chances of fertilization increase with more number of cells. Hence, instead of 2, making 4 cells is preferable in nature. This is the reason why meiosis gives us 4 daughter cells with half the number of chromosomes. Now, since the process yields us cells with half the chromosome number, it's also called as reductional division. Now that we've got introduced to the process, let's glance at the definition once more but this time with respect to the chromosome number. Mitosis is simply a process of cell division which produces two new identical daughter cells with the same amount of genetic material. So are these two daughter cells exactly similar to each other? Yes, they are genetically similar to each other. And this is the reason why mitosis is also called equational division. It's extremely important to maintain the amount of genetic material in any cell because uneven number of chromosomes in a cell can lead to abnormalities like cancer. Now, before moving ahead with the concept of mitosis, let's get through with a few important terms first. We have something called the DNA, chromatin, and chromosomes. So let's find out what exactly these concepts are. We've seen these tangled thread-like structures in the nucleus. Any idea what they are? Well, these are called chromatin. If we zoom in further, we find these thread-like structures appear to have this bead-on string arrangement. These beads are nothing but octamers made up of histone proteins. And what about this string? Well, that's our DNA molecule. Yes, this string-like structure wrapped around the histone octamer is DNA. And why is this wrapped in this fashion? That's because it helps in increasing the compactness of the complete structure. The wrapping reduces the volume occupied by the long stretch of the DNA, keeping its length intact. So it's the DNA which, along with the proteins, forms the chromatin structure. And what is this chromosome now? In any metabolically active cell, the genetic material is always present in the form of these tangled threads, that is the chromatin. But during cell division, these threads condense to form a structure which appears somewhat like this. And this is nothing but the chromosome structure. So is chromatin and chromosome the same? Technically, yes. The only difference is that the chromatin structure is generally found in a metabolically active cell in the form of tangled threads, while the same threads condense to form the classical higher order structure of chromosomes. So we can say that the DNA with histone proteins make up the chromatin, while the same chromatin condenses to form chromosomes. There is one more term that we need to know of. It's called the chromatid. Now what could that be? This is a single chromosome. Now can you tell me whether this is also one chromosome or half? It's also a complete chromosome. In both these structures, these arm-like structures which are attached to the central bulge are called chromatids. In simple words, the thread-like strands which are attached to this centromere are called chromatids. Now in this chromosome, there's only a single chromatid, while this one has two sister chromatids attached at this common point. Two sister chromatids symbolize the duplicated DNA. That means the DNA which is duplicated for cell division is attached to its paternal copy before the actual division.